In this video, we are going to talk about genetics of wine. So before starting this video, like this video and subscribe to our YouTube channel for future updates. The grape is one of the most established and most significant green yields. Grape and wine smell has for some time been of social and logical interest. The different compound classes involving smell result from numerous biosynthetic pathways. The genomic assets that are accessible to the grapevine research local area have expanded hugely during the beyond 5 years, in lined up with a restored interest in grapevine, Vitis vinifera L, germplasm assets and examination of hereditary variety in grapes. Hereditary variety, either regular or instigated, is priceless for crop improvement and understanding quality capability, and the equivalent is valid for the grapevine. The set of experiences in grape plantation social practices have generally resolved the hereditary variety that exists today in grapevines. Deep genetic evaluation. The grape is the most important green harvest on the planet. The organic product from the world's 8 million hectares of grape plantation is for the most part handled into wine, yet some is bound for new utilization as table grapes, dried into raisins, handled into non-alcoholic squeeze, and refined into spirits. The archaeological record recommends that development of the tamed grape, Vitis vinifera subsp. Vinifera, started 6,000 to 8,000 Y back in the Near East from its wild forebear, Vitis vinifera subsp. Sylvestris. The large number of grape cultivars being used today have been created from that point forward by vegetative spread and by crosses. Wine and table grapes presently get serious synthetic applications to battle extreme microbe pressures. This vulnerability to illness, in any case, isn't owing to an absence of hereditary variety. Vinifera harbors levels of hereditary variety a significant degree more prominent than people and is equivalent in variety to maize, with polymorphism that goes back huge number of years. Subsequently, an earth-feasible grape developing industry will depend on getting to and utilizing the grape's colossal hereditary variety to foster superior illness-safe grape cultivars through marker-helped rearing. Generally, grape rearing projects have looked for genotype aggregate affiliations utilizing linkage planning. On account of the grape's long age time, by and large 3Y, in any case, laying out and keeping up with linkage planning populaces is tedious and costly. In this manner, expansive affiliation, GWA, and genomic choice, GS, are appealing options in contrast to conventional linkage planning in the grape and other seemingly perpetual lasting natural product crops. Family analysis within vinifera. We utilized the Vitis 9K SNP exhibit to create 5,387 SNP genotypes from 950 vinifera promotions, 451 table grape promotions, 469 wine grape promotions, and 30 promotions of obscure sort, from the grape germplasm assortment of the USDA, one of the most extensive storehouses of grape variety on the planet. As of now, there are more than 10,000 grape cultivar names being used around the world, and their grouping is frequently confounding a result of homonyms, equivalent words, scant or wrong verifiable data, and curation mistake. A few cultivars have been separated into a few through the vegetative proliferation of substantial freaks, and we expect clones got from a similar cultivar to be hereditarily indistinguishable at the tried marker loci. We view that as 551, 58%, of the 950 vinifera promotions are clones of without a doubt one other promotion in the USDA grape germplasm assortment. Numerous clonal connections are confined to sets of promotions, yet gatherings of up to 17 increases were viewed as clonally related. We discovered that there are 583 special vinifera cultivars in the USDA grape germplasm assortment, 399 promotions with no clonal connections and an extra 184 increases made out of one increase from each arrangement of clones. Wines and table grapes exist thanks to a hereditary trait so uncommon that it's just happened 2 times in nature in the last 6 million years. Furthermore, since the taming of the grapevine a long time back, rearing has kept on being a bet. At the point when the present cultivators develop new assortments, attempting to deliver better tasting and more illness-safe grapes, it requires 2 to 4 years for raisers to realize whether they have the hereditary elements for the ideal bloom. Today, most developed grapevines are bisexual, while all wild individuals from the Vitis class have just male or female blossoms. As reproducers attempt to integrate sickness obstruction qualities from wild species into new rearing lines, the capacity to evaluate seedlings for blossom sex has become progressively significant. Furthermore, since grape sex not set in stone from seeds alone, reproducers invest a ton of energy and assets raising plants, just to dispose of them quite a long while down the line after learning their single-sex assortments. In the review, the group analyzed the DNA successions of many wild and tamed grapevine genomes to distinguish the novel sex-deciding locales for male, female, and bisexual species. They followed the current bisexual DNA back to 2 separate recombination occasions, happening somewhere close to a long time back. Many wine grapes can be followed back to either the first or second occasion genetic stock. Cultivars, for example, Cabernet Franc, Cabernet Sauvignon, Merlot and Thompson Seedless are all from the primary genetic stock. The Pinot family, Sauvignon Blanc and Gamay Noir begin from the subsequent genetic stock. What makes Chardonnay and Riesling special is that they convey qualities from the 2 occasions. Londo said this shows that old viticulturalists crossed grapes between the 2 genetic supplies, which made a portion of the present most significant cultivars. Reporting the hereditary markers for recognizing male, female and amazing bloom types will at last assist with speeding cultivar advancement and lessen the expenses of rearing projects. Financing for this study was given by a Specialty Crop Research Initiative competitive grant from the USDA National Institute of Food and Agriculture. Grape fragrance compounds. The Prevail compounds adding to the smell profile of grape berries fall into the accompanying classifications: mono and sesquiterpenes, methoxypyrazines, furan subordinates, lipoxygenase pathway items, and phenylpropanoid pathway items. 2 classes of mixtures, norisoprenoids and unstable sulfur compounds, are significant in wine yet are for the most part found as non-volatile antecedents in berries and require further adjustments to be perceived 2, 3, 11, 21, 26. During winemaking and resulting maturing processes, unpredictable mixtures and their forerunners can go through protein-catalyzed alterations and unconstrained synthetic changes. Further compound variety is presented by wine microorganisms and maturing vessels. This intricacy, alongside incredibly low centralizations of certain mixtures, huge natural and transient impacts on unstable fixation and creation, and formation of unpredictable mixtures into non-volatile forerunners has made examination into the smell parts of grape berries challenging 11, 21, 27. The trouble is intensified in the portrayal of qualities encoding proteins that produce fragrance compounds or their forerunners. In the resulting areas of this audit, we sum up the ongoing information in regards to the significant gatherings of grape fragrance mixtures and qualities engaged with their biosynthesis. Unstable sulfur compounds. Unstable sulfur intensifies territory from strongly rank to enjoyably fruity and are necessary to the smells of many plants, for example, passionfruit, dark currant, garlic, and asparagus. They are likewise significant unstable parts of meat, mushrooms, and wine 11, 21, 178. Until the disclosure of thiols that contribute positive fragrances to wines, sulfur compounds were for the most part connected with wine defects 178. The most widely recognized unpredictable sulfur intensifies in wine are thiols, hydrogen sulfide, dimethylmercaptans, and methylthioesters. Large numbers of these are the results of microbial digestion during aging. In any case, polyfunctional thiols and methylthioesters are pivotal parts of varietal character in certain assortments and begin from forerunners tracked down in the berry. Sauvignon Blanc is generally notable for fragrance characters got from thiols, however sulfur compounds are likewise significant. Phenylpropanoids. The phenylpropanoid pathway is notable in grapes as the beginning stage for the biosynthesis of flavonoids, anthocyanins, stilbenes, hydroxycinnamates, and other significant compounds 209-210. Less all-around considered is the biosynthesis of unstable phenylpropanoids notwithstanding their overflow in the glycosidically bound part of unpredictable mixtures in grape juice 11. As with lipoxygenase pathway compounds, grape monoterpenol glycosyl transferases have been displayed to create glucosides with phenylpropanoid compounds as substrates. In any case, it isn't known the amount they add to the pool of bound phenylpropanoids in the berry 72. Collectively, phenylpropanoid-determined unstable mixtures contain up to 20% of unpredictable aglycones in enzymatically treated Chardonnay juice, yet individual mixtures for the most part don't surpass discovery limit. What do you think about this video? Do let us know in the comments section below. If you enjoyed this video and want to hear from us again, be sure to hit that subscribe button before you go.